Good afternoon, y'all. This is Renee Kozishki giving you some information that correlates with Chapter 8, which is on metabolism. You can see here I've broken it down into main ideas. And over here to the side, I put the slide numbers in the slides presentation that this information corresponds to. There's a lot of good slides in that presentation, but it is long. And I'd hate for you to be there, sitting there, thumbing through every single bit of it. So if you want to just make life easier for you, the slides that are noted here in the margins, those are the ones that I would pay attention to. Maybe ignore the others. All right. Main idea number 1. Life is either building itself up or breaking itself down. When it builds itself up, that's an anabolic reaction. When it breaks itself down, that's a catabolic reaction. And so just like a pile of Legos that you would build up and break down, and then the next day build something new, that's how living things work. But the Lego pieces they're using are biological molecules like carbohydrates, lipids, proteins, nucleic acids. The classic examples of catabolic reactions are these two. Classic examples of anabolic reactions are these two. Now, what's important in this chapter is especially that you understand how these two are linked. Because anabolic reactions, they won't happen by themselves. They're uphill energetically. They create more order, but they require energy to even think about getting started, right? Exergonic reactions release an overall output of energy. And so what we do is, as living things, we use exergonic reactions to power endergonic ones. And this is a topic called energy coupling. Now, if you're in class, I put Gabe and Tim. One was on a trampoline, one was in a tree. And the goal was, the one in the tree, let's just say it was Gabe. He had to jump out of the tree, land on the trampoline in order to get Tim off the trampoline. This is energy coupling. Gabe coming out of the tree, bouncing down on the trampoline is exergonic. That's used to power Tim sitting on the trampoline, bouncing up as soon as Gabe hits that. So when that is in a cell / like scientific situation, we call that phosphorylation. So phosphorylation occurs when an inactive substrate becomes active because an ATP molecule transfers a phosphate group to the inactive substrate, making it active. And you can see that often comes with what we call a conformational change, which is just a shape change. So the inactive substrate is now active, changed its shape because a phosphate group from ATP was added. And now this is going to be much more likely to react. Because with this phosphate group attached, it's more unstable. So this be like Tim sitting in the middle of the trampoline, not moving. This be like Tim bouncing up. Why is he bouncing up? Because Gabe jumped down. All right? So hopefully that memory sticks in your heads of what we did in class. But the only other thing I'd add to this is that the enzyme that does that is called a protein kinase. Kind of a weird name, but a protein kinase, is an enzyme that takes a phosphate off of ATP and puts it on an inactive substrate to make it active. Now, there's other kinds of phosphorylation that you're going to see, but this would be called like energy coupling reaction, because what we've done is we've taken the hydrolysis of this bond, which is energy releasing, and we've coupled it to an endergonic reaction of changing the inactive substrate to an active one. More on that later. I'll elaborate more on phosphorylation next week, but hopefully that was a good reminder. Now, energy coupling by ATP, meaning primary energy source, the hydrolysis of that third bond is going to be coupled to lots of endergonic reactions in cells. Energy coupling is going to be used to power three kinds of cell work. Chemical work, endergonic reactions. Any chemical reaction that's endergonic going to be coupled to ATP hydrolysis, or basically just the transfer of a phosphate to an inactive substrate to an active one. All right, mechanical, any sort of movement. Like muscle cells, they move. Chromosome movement. That requires mechanical energy because we're moving chromosomes during cell division. Whole cell movement, or like cilia, flagella. Those structures move. They move because of mechanical work being supplied by ATP. And then there's transport work. So this is like active transport. You know what that is because you're so smart. Moving substances from an area low to high concentration against the gradient. Kind of like using a pump. Requires energy. It's transport work. Also, endoexocytosis. Example of transport work. All right. So, moving kind of quickly, jumping through the topics, but hopefully hitting the most important things for you. The next part of the chapter, corresponding to slides 32, 38, is enzymes. They're the ones that are like workhorses. They're working full-time in a cell, organizing cell work, organizing chemical work, organizing mechanical work, organizing transport work. What are enzymes? Type of protein. What do we know about that? Well, the most important thing about proteins is that they have a specific 3D shape. And that shape ends up creating active sites. And what fits into the active sites? Substrates. So, how do enzymes organize cell work? Well, they orient substrates in the right position. Substrates aren't just going to bump into each other randomly and shit. They're going to have to sit down and connect in the right orientation. All right, nothing they do. They weaken the substrate bonds. They make the substrate, let's just say, like, here's substrate A. It's not going to turn out well, y'all. Here's substrate B. I'm just saying the picture's not very well planned. Say this is a monosaccharide, this is a monosaccharide. Combining by dehydration synthesis, make a disaccharide. They're going to weaken the substrate bonds, the bonds in between these two monosaccharides, to encourage them to join with less energy. They allow cells to coordinate the frequency, the location, when, where, why, and how the reactions are happening. So enzymes organize all cell work. One thing that you should know is that enzymes lower activation energy. This is energy subscript of activation. How do they do that? They weaken substrate bonds. They position those substrates in the correct position while they are reacting. They make a meeting place and an energetically possible reaction to occur. So if this is the energy of the reaction, and this is time, you add an enzyme, there's less of a hill to climb to get that reaction going. The hill is shorter. We call the difference between the energy of, sorry, my bad, not that one. Difference in the energy of the reactants and the energy of the products, we call that difference delta G. All right, check your class notes. The ones we took last semester, they're going to be a little bit better than this. It's kind of, you know, sloppy, and it's not poorly planned. I'm going to say it's very successfully planned and executed. It's just kind of messy. So you can also check the slides for that. That'd be good. All right. Number 4, enzymes function best, which means the reaction rate is the highest at a specific temperature, pH. And here's another thing, folks, we didn't really talk about, but they're also sensitive to salinity changes, which means salt concentration. So what that means is if I take an enzyme that's used to working at a specific temperature and pH and salinity, and I change the temperature drastically, change the pH drastically or salinity, I can denature that enzyme. So this is like an unravel of the 3D shape. 3D shape is lost. Think y'all remember Beano? Remember that, Jenna Rhodes? Bean juice. Do that again on your own time if you want. You can get Beano at the pharmacy and beans at your regular grocery store. Carry on a denaturing lab. You can acidify the beans / Beano with some vinegar, lemon juice. Make yourself into a at-home scientist. A remote scientist, right? You guys are already that anyway. All right, back to the plan here. Enzymes max out or plateau at a reaction rate once all active sites are occupied. So here, as we increase substrate concentration, what are the substrates? The molecules the enzymes are joining, y'all. As we increase substrate concentration, we're going to increase reaction rate, increase reaction rate, increase reaction rate. Oh, what's happening? Reaction's slowing down. Why? All the enzymes' active sites are occupied. And then we get this plateau. The plateau is sometimes referred to as saturation. You can imagine that if my hands are an enzyme and this is my reaction, I got to join these two. There's going to be a rate at which, if you increase the frequency at which these are being added, sorry, I thought I had another one. Oh, here it is. I'm going to move faster, but I will only move as fast as I can as long as my active sites are occupied. All right, 2 more for this video, then we're done. All right, slide 46 talks about inhibitors. They're inhibited by toxins. They're also inhibited naturally / effectively, like on purpose. All right, 2 kinds of inhibitors. Competitive inhibitors block the active site. Oh no, oh, almost saved it. Blocking the active site. Ugliest picture you ever seen of an enzyme inhibitor. But if you look in the slides, see, saved it. You're going to get a better one. They block the active site. So the substrate can't bind. This takes the enzyme out of commission. Can't work. Allosteric inhibitors change active site shape, but they bind at a location that is not connected to the active site. So these inhibitors, they bind here, perhaps, and then that induces a shape change in the active site, which then blocks substrate binding. All right, so more on that, slide 46. I would study that one. I don't think this is going to show up too complex on your AP exam coming in May, but I do think these are very important concepts to review. Last, pathways in cells are regulated by feedback inhibition. Excellent image on slide 58. What this is when the product of a pathway, let's say the final Man, getting confused about my letters now. Final product of a pathway, allosterically inhibits the first enzyme in the pathway. What's the advantage of this? Well, evolutionary speaking, organisms that can shut down pathways that aren't necessary are going to have an advantage because they conserve energy. So if I'm making cookies and the cookie's the final product of my work, my pathway, which involves all sorts of steps, what if actually having the cookies in my hand inhibited me from making more cookies? That'd be great. All right, y'all. Ask me any questions you want. I know this is ridiculous. Having a little bit of fun. Miss ya. And that was 16 minutes, chapter 8. Bye.